All right, everyone. It is 5:50, so we will get started with our faculty speaker. It is our pleasure to introduce Dr. Vora. Dr. Vora is a maternal fetal medicine geneticist at UNC. She comes to us from her OB-GYN residency at Tufts Medical Center, where she also completed a fellowship in maternal fetal medicine, specializing in clinical genetics and high-risk pregnancies. She moved to UNC in 2012, and just, well, I guess 2 years ago now in 2021, she was awarded an R01, a top-level NIH grant to study genes critical to human brain development. She was interviewed by NPR last October on today's topic, and we are incredibly excited and honored to have her speak with us today. So we'll turn it over to you, and thank you very much.
I'd like to thank the organizers for inviting me to speak today, and also like to thank the audience, also the virtual audience. I think it's really great that people come out and hear some of these topics. So Amber did a really great job. I was just telling her, of explaining, kind of giving a background on what we do, which is prenatal genetic testing. And so, you know, as you introduced me as a prenatal geneticist, what I do really is I take care of families where the pregnancy has a birth defect. And so my husband kind of describes me as the kind of OBGYN you would never want to have to see. But, you know, to the other audience member virtually who asked, how has genetics evolved, prenatal genetics evolved over time? I mean, in the last decade, things have changed so rapidly that what I did in fellowship, we're no longer even doing because it's become obsolete. So it is a very exciting time to be in genetics and in prenatal genetics especially. And so on this slide, I put an article that was in the Huffington Post about, you know, how to really understand what a prenatal genetic test results actually mean, because you know, there are a lot of tests that are available. And, you know, and this article has explained as well that the testing has rapidly changed in recent years. And so there's a lot more information that families have to deal with than they had to deal with before. And so this article kind of goes over how to understand what some of these prenatal genetic tests actually mean. And I'm going to attempt to talk about some of this in the next 30 minutes and hopefully leave time for questions as well. So, you know, when you get pregnant, everyone tells you, you know, do this, do that, take a prenatal vitamin, don't smoke, don't drink alcohol. And you could do everything perfectly right, get your immunizations, go to your doctor, you know, manage all your medical conditions. But as Amber said, you know, there's birth defects that affect 3 to 4% of all pregnancies. And even if you do everything perfectly right, there's still a chance, a small chance, 3 to 4%, that you can have a fetus diagnosed in utero with a birth defect. And so that's where I would come into play as a clinician that you may meet with after an ultrasound shows that there is a problem. And, you know, it is hard sometimes because as a prenatal diagnostician and geneticist, you know, families come in expecting to find out the gender, is this a boy or a girl? And then, you know, what we're doing is we're screening for birth defects. And that's the whole purpose of the ultrasound done between 18 to 20 weeks, is we're looking at the brain, we're looking at the heart, we're looking at the kidneys and the organs, the spine. And because we're screening for those, that, you know, we do find the birth defects. And because we're a big tertiary referral center, we do see a lot of families affected with a birth defect. And so this, you know, can also be hard when you happen to see a lot of birth defects, because like, for instance, in my own pregnancy, most pregnancies are healthy and normal, but, you know, you're skewed with what you do. And so, you know, my pregnancy, I was basically scanning myself every day, like what's in there. And, you know, Amber went over a lot of this already, but basically these are the slides showing the timing of genetic testing. So even before implantation, there is an option that some families can have to they would have to go through in vitro fertilization, which is where eggs are removed from the ovary and combined with the sperm in a petri dish, creating an embryo. And you may have heard of IVF. This is for women who may have infertility issues. And when this embryo is at the 8-cell stage, they can take one cell out and screen for genetic conditions. So that can be done even before the person is pregnant. They can test the embryo, and then if the embryo doesn't have a genetic condition, they can then put that embryo or transfer that embryo into the uterus to then hopefully have implantation and then a healthy pregnancy. So that's the earliest. And then Amber went over the other options called chorionic villus sampling. That's the biopsy of the placenta, and also amniocentesis. That's where we put a needle in, take fluid out, and send that fluid to the lab to check the chromosomes. Those are two tests that we do very commonly here in Chapel Hill. And, we get referred in for people who need these tests. And so those are pretty common. Those do have a small risk of miscarriage with the test. The CVS, or the chorionic villus sampling, is a 1 in 500 risk. It's less than 1%. The amnio is a 1 in 1,000 risk. And then when the baby's born, then every baby in North Carolina gets what's called a newborn screen. So they screen the baby for lots of different conditions that are medically actionable, that can be treated after birth. The baby also gets a hearing test, because some babies are born with hearing loss that can be genetic. And then, we always encourage all reproductive age women and men who are planning to have a family to have carrier screening. So you may have heard of a condition, for instance, called cystic fibrosis or sickle cell anemia. We're all carriers of 4 to 5 conditions that if we're carriers and we have a baby with someone who also is a carrier for the same condition, then we could have a 25% chance of having a baby affected with the condition. But when you're carriers, you're not affected. So these are the kind of the pictures showing, as you can see, the needle going into the uterus, into the sac of water around the baby, that's called around the fetus, that's called the amniotic fluid, that's called the amniocentesis. And we do it all under ultrasound guidance. So, and then the same thing with the CVS, we oftentimes put a catheter in through the cervix. So it's going in through the vagina, into the cervix, and into the uterus to obtain that piece of placenta. And we send those off to the lab. So I put this slide in here because, I think unless we're living under a rock, we all know what's going on in the country about abortion. And the reason I put this in was because, like I said, we do our anatomy surveys between 18 to 20 weeks. And that's kind of the earliest time where we could make a diagnosis of birth defect. And in North Carolina, we are allowed to do terminations up to 20 weeks. And we follow the guidance from the state legislature at UNC. And if a patient wants to stop the pregnancy after 20 weeks, then they would have to go to a different state. However, when we find a birth defect, and as Amber showed, there are birth defects where the baby can't survive outside the uterus, and also poses a risk to maternal health. And so, I think it was just important to bring this up, that really in this particular patient, she talks very poignantly about having to make the most difficult choice of her life, which we see all the time with our patients. It's a desired pregnancy. There's a condition that is life-limiting in the fetus. And unfortunately, diagnosis was made pretty late for her state, and then she had to, travel out of state. So I think this is something that, we need to think about in terms of prenatal diagnosis. So basically, as I said, when I was a fellow, we used to when there was a birth defect, we used to send a karyotype. A karyotype is basically looking at are there 46 chromosomes? So, I know I have 46 chromosomes because when I was a fellow, I did my own karyotype. And I assume everyone in here has 46 chromosomes, but we get 23 from our mom, 23 from the dad. And the standard karyotype can only tell you really if there's an extra chromosome, like you may have heard of Down syndrome or trisomy 21, or a missing chromosome. It can't really tell you anything that's higher resolution than that. So it's a very rudimentary test that can only really screen for some conditions, but really not very many. A microarray is a test that came about in 2012. There's a big study that showed that the microarray had a better diagnostic yield than the karyotype. And so microarray can find smaller deletions and smaller duplications or extra pieces of chromosomes that the karyotype could not find. So for instance, if you had, and you can remember base pairs, if you're missing 10 million base pairs, the karyotype would probably find that. If you're missing 50 to 100,000 base pairs, then the microarray would find that. Now, if a fetus has a birth defect, we're sending a microarray. That's standard genetic testing. But the microarray is normal, then what? Well, we started sequencing the DNA of the fetus. And DNA is in the nucleus of the cell. We're made up of billions of cells. So you may have heard of like people explaining DNA, like you take out the DNA of every cell in your body and you line it all up, it would be like 67 billion miles long. So it's amazing. So what we do is we sequence the DNA. And so you may remember from your biology classes that their base pairs, A, T, G, and C stand for, adenine, thymine, guanine, cysteine. Sequence all the base pairs and then look for mutations or single variants that can cause a genetic disorder. Okay, so this is the cutting edge technology that is now being applied to prenatal diagnosis. So what I'm going to talk about is just actually obtaining fetal DNA directly from either an amniocentesis or a chorionic villus sampling. And then in patients with birth defects, those patients have a normal microarray and they went on to enroll in our study. So this is, we've now sequenced 340 cases of birth defects, and we found a result in about 28% of those. And then I found no result in 72% of those. And I'm going to tell you at the end, if we get to it, what we're doing for those 72% of families that don't have a result. So finding the result, as Amber said, helps with recurrence risk counseling. It helps the family understand what is causing the birth defects. It helps the neonatologists or the doctors that take care of the baby after birth prepare for what's coming when they know that there's a baby with a certain genetic condition coming into the NICU. They know how to prepare and optimize the care of that newborn. It also allows for pregnancy decisions. And we're also doing research showing that patients who get a diagnosis, they cope better in the long term from knowing that information. And so these are some of the types of diagnoses that we find, and I'm not gonna, go through each one of them, but these are rare genetic conditions. And so that's just one thing I wanted to emphasize, is that most pregnancies are healthy and result in, a healthy baby, but this talk is very skewed towards rare diseases. And so all of these things would present with an abnormal ultrasound finding and then result in an amniocentesis if the patient chooses that. And then we would do the sequencing and find an answer. So sometimes it's a new mutation in the fetus. That means neither parent has it. It wasn't passed down from one parent to the fetus. It's just occurred. It's de novo or new. Other times it's what I explained before, that both parents are carriers for a condition and they have a baby together and then the baby has the condition. So these are just, again, some of the conditions, and some of them, if you know ahead of time, then you can start treating the baby as soon as the baby comes out. So that's another benefit of knowing ahead of time. Now, rare diseases are a group of disorders that have a low prevalence overall. but there are 260 to 450 million people affected by a rare disorder. And we all have around 20,000 genes, but we really only know what around 6,000 of those genes do. So there's still a lot to discover in humans. And a lot of the rare disorders that we see do affect the child, and 90% of pediatric rare disorders affect the nervous system. So it takes a long time for a patient with a rare disease to get diagnosed, at least it used to. But now we have sequencing technology. So as I showed you, we can sequence the whole genome. And so it's still hard to get these life-altering genetic tests. And because a lot of it is insurance, a lot of it is cost, a lot of it is, patient going to see doctors who may not be aware of these sequencing technologies. And, there are a lot of people on Earth who suffer from a rare disease, and a lot of those rare diseases are genetic. And whole genome sequencing can provide a diagnosis in about 40% of cases. And then, we just need to try to figure out as we move forward, how can we make access to these technologies equitable? So this was a story that was in the news that actually got picked up from Facebook. So there was a physician at Beth Israel Deaconess Medical Center in Boston, and there's a Facebook group called Physician Mom Facebook group, which actually a lot of people talk about this group. I'm not on Facebook, but a lot of people really find out a lot of information from this group. And there was a baby that was born with a lot of developmental abnormalities. She wasn't meeting her milestones. And so after reading this post, this woman told her husband, who was a neurogeneticist at Harvard and doing sequencing on genomes, and he said, let's sequence this girl's genome. So they sequenced the genome, they found the variant or the mutation that caused the problem in this girl, and then they were able to design a drug that was gene therapy for this girl, and her milestones have improved. So this is just kind of an example of like, how can sequencing improve health conditions for patients with rare disorders? Is this good? Can I play that? Next? Oh, I'm gonna play this video. Is it fine?
For 30 years, I was limited. I could never do things myself. Just pumping gas and just getting out of the car and pumping my own gas. I bet a lot of people hate pumping their own gas. It's little to regular people, but big to me. It was really big. When I was around 6 years old, I started walking on my tippy toes. And as I got older, it got worse. Probably around 6th or 7th grade, I started using crutches all the time. Or I remember kids making fun of me on the bus. And then at school, I didn't even want to go to school. I've had the foot surgeries in Duke, 5 on one foot and 5 on the other. And then, like medicine, taking them and thinking, oh, this might help me, But it didn't work.
In genetics, we're used to seeing people who have been to doctor after doctor, year after year, and not getting a diagnosis. That's the diagnostic odyssey. You can imagine that's a very frustrating process for patients.
Through 7th grade till I was 20, I didn't go in places a lot. And if I did, I used my crutches. I just wanted to get better. I wanted to do things. I wanted to live a life. I didn't have a life.
It's not a coincidence that we're at a public institution. That's important because we have this amazing new ability to sequence DNA. And what we're investigating is how we can use it as a diagnostic tool. How can we use that in patient care? So the name of the project that we're focused on at the moment is called NC Genes. When we started this project, we said to clinicians, if you have patients who you haven't been able to diagnose, but you think that maybe there's something going on genetic, we'd like them for our study. A great neurologist with whom I've worked for years named Jane Fan told us about a patient of hers who, because of her age of onset being young, suggested maybe this was genetic.
I was scared, to have my genetics tested. But then I thought, well, if they find something, I just don't want it to be worse. If I can just stay with crutches, just don't let me get no worse.
Our ability to sequence DNA has undergone a dramatic revolution in the last decade. We can now do 1 million reactions at the same time, and we can find out your entire genome. In this particular case, Ms. Davis, with the help of the computer, we sifted through these variants, and one of them kind of stood out. And this gene had been associated in the medical literature with a condition called dopa-responsive dystonia. And dopa is a very commonly used drug. It's used as a first-line treatment in anybody with Parkinson's disease.
By the time I started taking my pill, it was 3 days later is when I felt my toes move. But I'll never forget it. Standing there on crutches and my toes wiggled, flattened out.
I swear, 6 weeks later, she comes back to our clinic and she left her crutches at home. She's walking, right? It was awesome. I mean, it's why you do research.
1 minute you're on crutches and the next minute you're not. So I wasn't sad to let them go, though. It made my life a lot better. A lot better.
We're very interested in figuring out how to use this technology to help the general population. Our mission really is to improve the health of North Carolinians. Scientific research is the way we improve our lives. It's a way to a better future for all of us.
I love that video. The man in the video with the DNA in his tie was actually, he was my mentor for my research. He has since retired, Jim Evans, but he's a wonderful man. And at UNC, they're doing a lot of research on adults and children in DNA sequencing, and our group is doing it on the fetus. So one of the things I wanted to bring up about the sequencing technology is that patients of different ancestries have a higher likelihood of receiving an ambiguous result from our sequencing data compared to patients of European ancestry. And there are historical reasons why patients of certain ethnicities or ancestries do not participate in research. There have been some historical abuses in the past, especially in African-American populations, and definitely a distrust of the medical system and enrolling in research studies, specifically using DNA. So we need to try to address this because there are some health disparities that are cropping up because of these new technologies, and we don't have enough information on the different pattern of variants in sequencing data for different ancestries. And so this is just a study that showed the reporting of race in genome and exome sequencing studies, and the bottom line is that the Black patients in the studies were far less represented, and there was much less data regarding their sequencing data, because we need population-specific references. And so we just have not had good luck in getting really good references to analyze their data as well. So the sequencing does clearly increase diagnosis, but which groups would benefit most? If we just start sequencing all healthy people, we would find so many different variants that may have no particular meaning and then cause a lot of anxiety. What's the clinical utility? Clinical utility is, what is done differently. If you get a positive diagnosis, does that improve your care? What is the change to your management? Can we develop a pipeline to deliver rapid results? As you can imagine, with prenatal sequencing, we need to be lickety split. We need to get those results to that family as soon as possible. Cost is a big issue. When I first started doing this, a genome was in thousands. That has decreased dramatically now. It's a couple $100s. And that's because we're doing more genomes. So the more testing we do, everything becomes cheaper. So eventually, I do think it's going to be something that can be accessible for population-wide screening potentially. And there are certain limitations of sequencing. It's still not perfect. Also, how do we handle incidental findings in the prenatal setting? So we could find a variant that could cause breast cancer in the fetus, and that could crop up when that fetus is 50 years old. So there's other ethical things that we deal with and discuss, and a lot of pre-test counseling and post-test counseling is needed. We have genetic counselors who help us with that. We're also doing some interviews called Qualitative Research, and speaking with families about their perceptions, their feelings, their psychological impact of the genetic testing that may have impacted them during the pregnancy. And we want to understand the ethical and social implications of applying prenatal sequencing in patients whose fetuses have a birth defect. And this is Dr. Asha Talati. She is one of my mentees, and her research really focuses on this aspect of what is the patient actually understanding that we're saying? How is the information impacting them? And it turns out that patients who enrolled in our studies had high hopes and expectations regarding the sequencing providing an answer for the fetal anomaly. And as I showed you in the pie chart, about 28% got a result. That still meant like 70% were not getting a result. And I also found that women who did get a result had higher test-related anxiety compared to those who don't. And so we're want to understand these factors because we want to support women who do receive results, and also optimize ways of providing this information. I think there's a big change in how people really process information. A lot of it is digital now. So a lot of decision aids are being developed and gaming apps on the phone, which you may like that or you may not like that, but that is what is happening now. And so we're also publishing on the ethical challenges in prenatal sequencing. And then, I just wanted to briefly talk about, remember I said that we only know about what about 6,000 of those genes do. We have 20,000 genes. So what we feel as a prenatal geneticist is that there are a lot of genes critical to human development that we don't know about. And so these are rare disorders, but when we do the sequencing, we have an inconclusive result. In my cohorts, about 72% of the time, but we still really look through that data and we look for changes in the DNA that could affect the protein structure that we think are highly suspicious to cause a problem in genes where we don't know what that gene does. We then put that gene and that mutation into something called Gene Mattress. So you could think of it almost like a dating app, but it's actually an app or a website for scientists studying genes, and it links you with another scientist who may be interested or looking at the same gene. And so it's called gene matcher. And so when you put your gene in, then you might get linked with other people. And in this way, we've found and kind of crowdsourced to find patients with the same constellation of findings with mutations in the same gene to then identify, okay, this gene may have a critical role in human development. And so when we find one of these genes, then, there are animal models, because we obviously can't, do gene editing in a human fetus. So we in our lab are using zebrafish. Zebrafish have 70% of the same genes that share with us humans, which may seem amazing to you, but it's actually really fascinating how the genes that we have are so highly conserved across species. And these genes that are highly conserved across different species are the genes that are very important. And so they're also can be easily modeled, and they're translucent, so you could see the development of the zebrafish as the zebrafish is developing, and they're really easy to kind of do experiments with. And so basically the way we do it is you collect zebrafish embryos, then you use a gene editing technology, and then you watch the zebrafish develop. If the zebrafish has the same kind of, let's say, brain abnormality that the fetus had, and you saw in the fetus there was a change in a gene that had a mutation that was very highly suspicious, then we knock down that gene in the zebrafish and we look at that zebrafish's brain. There are brain abnormalities, then we say, okay, this gene seems to have a really important function in brain development. And so that's how we use the animal model. And so this is just a couple pictures showing that, there are changes in the cerebellum, which is the back of the brain that helps us with movement and balance in the zebrafish. And this was a case where the fetus had a severe cerebellar abnormality. So the phenotype or the manifestation of the problem in the fetus was recapitulated in the zebrafish. And these are two genes that we found were important for human brain development. This was a gene that is important in a lot of different things, but it's a gene that we saw were abnormalities with the heart, the brain, craniofacial structure of the fetus, the kidneys, and all of those were recapitulated in the zebrafish. And these are just pictures you can see of zebrafish. And so there are in utero treatments. So if we understand the genetic condition in the fetus, there are starting to become treatments in utero where you can put a needle into the umbilical cord and put stem cells in or put gene therapies in to try to improve the outcomes for the baby even before it comes out. So that is what I have. Obviously, there's tons of people behind this research. It's not just me, but mainly grateful to the patients who enroll in these studies in a very difficult time. Obviously in their lives, the funding sources, and then Kelly is my study coordinator, and she's really the brains behind all the operations here. So thank you, and I'll be happy to take any questions. So the question was, how does pre-implantation genetic testing differ from prenatal testing? And that is a good question. So as you can imagine, when you take a cell out of an 8-cell embryo, you can screen for some things, but you're not really able to do whole genome sequencing on that one cell. You could, but there could be some false results that, since you're only taking one cell. And so that, definitely is something that is not being done. And there's also a chance of getting false negative results. So we always suggest, and it's recommended by the major societies, that if you have pre-implantation genetic testing, then you have either a CVS or an amnio to confirm that that fetus is unaffected with that condition, because it's very, you know, facile manipulation of one tiny embryo, and so mistakes can happen. And so it's not the most comprehensive type of testing. Yes. Yeah, that is a very good question. And we do know that patients who have IVF, pre-implantation genetic testing, there are certain pregnancy specific conditions that are more common in those patients. And so we don't know if it's the infertility itself. We don't know if it's the procedure or what exactly it is that, you know, is causing those increases in pregnancy specific complications. I think that, you know, they have done studies, and I think that most of the time things go fine, but certainly that is a concern that has been raised. Mhm. Yes. So ethically, you know, I think the ethics community is very against it. And then biologically, I think there's still a lot of problems. There can be a lot of off-target effects. And in China, you may have heard about it in the news, they did try to do some gene editing in human fetuses. And, you know, I think there were a lot of off-target effects. So we're currently not doing anything like that. Now, people are talking about things like that, but it's kind of at this point a theoretical discussion, and probably will need to involve an international board of some sort of ethicists.
What stage do people?
Mhm. Mhm. Yeah, that's a good question. So actually, when Amber put her slide up, that the average age, that gestational age that, you know, pregnant women find out they're pregnant, 5.5 weeks, is, you know, for our patient population, that's very early, because we take care of a lot of patients who have some of those characteristics that you had mentioned in your slide that the study showed, that come late to prenatal care. They have a lot of barriers to healthcare. And we are often finding these diagnoses late in gestation. And, you know, we are a big tertiary care center. So for instance, you know, I think we probably diagnose, you know, even 2 to 3 anomalies a day in our ultrasound unit. Some of those are referred in. Some of those are new diagnoses that just basically, you know, family comes in thinking everything's fine, and then you have to drop a bomb on them. And so, you know, we have 3 busy prenatal diagnosis units, Chapel Hill and Raleigh, and then another one in Chapel Hill. So there's 3 different ones all going on every day, Monday through Friday. And each one of those has a couple anomalies a day. So it is something we do see a lot, but we're also trained in diagnosing these birth defects.
We have another question. In what ways has Neeta been involved? And how has changes to
That's a good question. So I think that, prenatal testing continues and we will continue. I think one of the ways that it's affected us is that, we have a 20-week ban here. And so we've shifted our ultrasounds to earlier in gestation. And, the fetal heart at 16 weeks is like the size of an acorn. So that's really hard to see like what's going on. And so also the average body mass index of our patients is around 30, which is considered overweight. And so it's really hard to penetrate and with the sound waves to see with the ultrasound. So I think that it's made it harder to find things early enough to allow patients to make a decision. I think that, we're going to still keep trying. And, people have talked about a 13-week ban. I think that would be really hard, obviously, to diagnose any kind of birth defects in a timely manner. I think that, in terms of designer babies, I guess it depends on what the person means. So like, when we do an amnio and we diagnose a genetic condition, that information is to provide the family with that information so they can make the best choice for themselves. We don't really have a way to, really do genetic testing for height and, eye color and things like that. And I don't think we should. I think that that's pretty far off in the horizon, but I know people sometimes mean that when they say designer. Now, there are also disability advocates, families who raise children with Down syndrome, and we work with those families too. And we give our patients, support group information. We provide the most up-to-date information on these genetic conditions. We, connect them with families who have raised kids with these conditions. And so, our job is definitely like, just give the information and give them the options available and let the family decide what's best for themselves. And so we work closely with the disability community as well. And, the disability community, they understand prenatal genetic testing, and, we're not trying to rid the world of babies with certain conditions. It's certainly not what we're trying to do at all. It's mainly providing information, which is actually information that patients really want. And that's why, a lot of these testing, the screening tests that Amber talked about and the testing that I'm doing has really exploded in the last decades because patients really want information. Mm-hmm. Mm-hmm.
If there is any way besides helping to do stigma trials,
Mm-hmm. Mm-hmm.
trials, any other way?
Yeah, thank you for that question. So the question was, can I think of any other ways to reduce disparities and, be more inclusive with patients of different backgrounds in terms of research studies and, whether they would enroll in those or not? I think, those are conversations that we're trying to have. We have a supplement to our R1, which is looking at this question and building focus groups, really trying to understand patients' perspectives from these different groups with different backgrounds and barriers to enrolling in these kind of studies. Because if you look at this cohort that we have, it's very highly educated patients who self-select in, patients who may find out about this option, they want sequencing, and this is free sequencing. So these patients also have a high, in our cohort, likelihood of having a college degree. And so I think it is a very self-selected cohort right now.
Right. Looks like there's no more questions in the chat. So we just want to say again, thank you so much for being here tonight.
Thank
Really
you.
appreciate both you and Amber's talks. I think we've all learned a lot. And to everyone in the audience, both in person and virtually, Nate and I, on behalf of the entire Mini Med School team, just want to say thank you again for attending our 4th year of Mini Med School. This was a great time on our end. We hope it was great for you too. Please be on the lookout in the next few weeks for a survey coming out to the listserv. We're going to try and get your thoughts on what went well, what didn't go well with our new hybrid format this year. And as always, any topic ideas or suggestions you have, we want to make this something that you guys are excited to attend. The other thing is Nate and I want to say a special thank you because this is our last year leading Mini Med School. It's been really great working with you all.
Yeah, I just wanted to say thank you to all of the audience members who have attended regularly and have participated in these sessions, as well as thank you to all of our speakers, both faculty and students. And thank you to all of the students who have helped put together this fairly large undertaking. And Kevin, you want to introduce our successor?
And just want to echo Nate, it really does take a village from the people who started this more than 4 years ago in the planning and all the people working on it now. So really our heartfelt thanks to everyone. And we are really excited to announce Paige as the new co-leader. You've seen her speak at some of these other sessions, and we leave you in her capable hands in the future. So we're excited to see what she does next year.
Thank you all very much for coming. And Paige, you want to say anything?
Excited to take over next year. I'm going to many suggestions. So any concerns, home offer.
I don't think anybody heard what she said. Please send your suggestions through the survey or to the UNC Mini Med School email address, which I believe is uncminimedschool@gmail.com.
Thank you
all again.
It's been great. Have a great night.